Okay, good afternoon, everybody. My name is Jerry Wells, and I'm going to be reporting on the progress in work package 5, which is about the influence of maternal diet on epigenetic programming in utero and offspring. And I've called this Microbes, Metabolites and Pig Health, because this is the key words which essentially capture the essence of what the talk is about. So in this work package, I should put this on presentation mode. Sorry. In this work package, yeah, we have performed a big gene bioenvironment study, and we are. Sorry, you can hear me, okay? Yeah.
Yes, no problem.
We generated the RNA-seq and ATAC-seq data from this study. And all the biorepository of samples, and we are busy with analysis of the data and the integration of the data, which will be necessary for the full interpretation. Okay, so diet, microbiota and health. I'm sure everybody knows that changes in the microbiota have been associated with many human diseases, even those that don't seem to be directly involved in the gut. So there's an interplay between the host and the microbiota, which affects disease susceptibility, disease severity, and also treatment resistance. So antibiotic use, for example, or infections in animals is something that can also cause this dysbiosis, and also diet. So diet, and particularly the lack of fermentable fiber, which reaches the colon, is one of the reasons why it's hypothesized that humans have had an increase. Our population has said in the Western societies in particular have had increasing occurrence of allergies, autoimmune disease, and also other diseases as well. So in the last decade, there's been a lot of research looking at diet microbiota research, and it's become very apparent that the metabolites produced by the microbiota, which is obviously connected with the diet, play a big role in human health. For example, some peptidoglycan fragments which cross the barrier are very important in enhancing systemic immunity and priming of macrophages and neutrophils, for example. The bottom one is a study about the aryl hydrocarbon receptor, which is a xenobiotic receptor, and metabolism of tryptophan generates metabolites that are recognized by this receptor. It's all kinds of functions in the host, depending on which cell types express the aryl hydrocarbon receptor, including gut barrier defense, including immunity, including immune regulation. And this is a study that we were involved in recently, showing the potential to use increased tryptophan in the diet in celiac patients to alleviate intestinal inflammation. At the start of the project, we wrote a review about the metabolites that we focus on in this project. At least we designed diets to deliver higher concentrations of these metabolites in the colon, and then in the circulation. And we are focusing on the short chain fatty acids. So just for definition, these are acetate, propionate, and butyrate. These are found in concentrations of up to 100 millimolar or more in the colon, in the feces. And usually acetate is about twice present at twice the concentration of propionate and butyrate. And I'll tell you more about levels in the circulation later. But one of the things that has become very clear, and a lot of these studies are in mice. There are some studies also in humans, in diabetic patients with short chain fatty acid supplements, and those people in the seminar that come from the nutrition area will know also there is a big interest in using this in animals, in agriculture. So short chain fatty acids per se in the diet. And in humans, they have been shown no, in mice, sorry, they've been shown to be important in epigenetic imprinting in bone marrow cells. So if you take germ-free mice and then you compare histone modifications in those with germ-free mice which have been recolonized with a normal fecal microbiota or have been just administered those 3 short-chain fatty acids, you see it almost recapitulates all of the epigenome effects that you see with a conventional microbiota. I won't say much more about these because we for time reasons we need to. But why short chain fatty acids? Just a few more papers from not in pigs but from the whole field. So in humans, in mouse models, acetate yielding diet imprints an antimicrobial program. So this could be important in infection. Acetate delivered in the drinking water has been shown to attenuate asthma in a mouse sensitization model. And it was also effective in the maternal diet. So that was just given in the maternal diet. That interested us a lot when we were writing this proposal, but also the short-chain fatty acids have been shown to prevent the onset of type 1 diabetes. So this is not the obesity-associated form of type 1 diabetes with metabolic syndrome, but the type 1 diabetes that is associated with some genetic factors as well as environment or lifestyle factors. So short-chain fatty acids then. So how do mammals get exposed to short-chain fatty acids, and what's the impact of the diet? Well, one thing that's happened in humans is that our fiber content in the diet, but not digestible fiber, but non-digestible fiber, has decreased dramatically compared to our ancestors several thousand years ago. And now it's considered relatively low compared to what it was then. And non-digestible fibers pass from the small intestine into the colon, and they're the more complex and higher numbers of microbes found in the colon are involved in fermentation of those polysaccharides. So they have many more enzymes to break down these complex carbohydrates. And as a byproduct of their metabolism, they produce short-chain fatty acids. And these are what you find in the colon at concentrations, as I said, of say in humans, around 60 millimolar per gram of content of acetate, about half that as propionate, and a bit less as butyrate. So in the intestine, short-chain fatty acids have been shown to have numerous roles in maintaining the gut barrier and interacting with the mucosal immune system there. So they can increase regulatory T cells, which helps control inflammation. They induce digestive hormone function through receptors on enteroendocrine cells. So these are a population of cells in the gut epithelium that produce hormones in response to metabolites. Short-chain fatty acids are one of those metabolites that can induce hormones such as PYY for appetite, or these others. Some of these are also involved in like GLP-2 in increasing the surface area for absorption. Butyrate importantly also serves as a fuel for enterocytes. And it's one of the reasons probably that it's not transported at such high concentrations into the lamina propria. And they have direct effects on epithelial cells, inducing many gut defense molecules. So what's going on in the animal field? There are already indications that short-chain fatty acids have quite profound effects in animals as well, and in pigs, which is the subject of Work Package 5 study. For example, the infusion of short-chain fatty acids in the ileum increased carcass traits like meat quality, lipid metabolism of growing pigs. And in a review, it was concluded that short-chain fatty acids could prevent fat deposition via regulating hormones and genes in these animals. So there is a bit more information also in pigs, but currently it's relatively sparse at the functional level. So these are sort of concentrations that you can find in serum and different parts of the intestinal content in pigs. And this comes from this paper I showed from Xiao et al. In Animal Nutrition. We've also measured them in feces on the pigs in different diets in work package 5, and we find similar levels. I'll tell you more about the differences and the effects of the diet in a moment. But in the colon, for example, acetic acid, 45 millimolar. This would be per liter of content, but if you in grams, it might be very similar to humans, about 60 millimolar per gram of content. Less propionic acid, and then butyric acid, about usually around 10 millimole per gram in our case, and also in humans. So quite similar to humans. There's much less short-chain fatty acids in the ileum, and notably some of these, like butyric acid, is very low, and propionic acid. So it's mostly acetate produced by those facultative aerobes that are involved in the digestion or in the fermentation and competition with the host for carbohydrates there. Okay, so a little bit of introduction now to the effects that we are expecting to see of diets that will influence short-chain fatty acid availability in pigs and epigenetics. So I'm sure people in this seminar are familiar with this, but DNA is wound up, compacted by histones, and these histones form a complex, and the acetylation of the protein tails or other kinds of modifications on these histones affect the compaction of the DNA, and that typically DNA which is more open and not compacted in these nucleosomes is more accessible and more active for gene expression. What this is just a summary of what I've said, that acetylation and methylation play important roles. Acetylation of the leads to more open chromatin and expression, whereas the deacetylation leads to more compaction. Butyrate and propionate are known to be inhibitors of class 1 histone deacetylase activity. I personally don't think they directly inhibit the histone deacetylases. They don't look like pharmacological inhibitors of these enzymes. I think it's more indirect, and perhaps through the AMP kinase or the other signaling mechanisms. Acetate is very likely to activate histone acetylases through effects on acetyl-CoA pool, which increases AMP kinase activity. And that's known, for example, to acetylate a number of histone deacetylases, but also histone acetylases. So that brings us to the aim of work package 5. So in this work package, we want to investigate the possible role of short-chain fatty acids as epigenetic regulators. And we focus on 2 different types of tissue, liver and muscle metabolism. The muscle makes a very strong link to the work I showed earlier, indicating that that infusion of acetate into the ileum, which is very different to what we do, had an effect on muscle mass and triglycerides in the blood and so on. So based on all this evidence and research that I've been telling you about, our hypotheses are that high fiber diets, which increase the concentration of acetate in particular, because butyrate is not taken up in very high amounts across the epithelium and going into the circulation, where it is the case for acetate, to some extent propionate as well. That these diets, through changes in the microbiota and fermentation, will increase the serum concentrations and the availability in the circulation to the piglets. We also wanted to explore whether these diets in the sows would have effects on the fetus in utero. And I'll show the plan for the experiment in a moment, because evidence in mice is that short-chain fatty acids in the circulation can exchange in the placenta and then have effects on disease outcomes. We hypothesize they would also have epigenetic effects, but that has to be still shown. We also designed the experiment with the idea that if there was effects of these diets in the sows, that there might be some imprinting that will have phenotypic effects in the offspring for at least 8 weeks postpartum. And this is because of studies in rodent models. Serum acetate, we predicted, might regulate PPAR alpha and lipid metabolism, reducing lipogenesis and lipid triglycerides in the blood, so leaner, but also affects on muscle directly, increasing muscle mass through upregulating type 1 myosin, and increasing the oxidative capacity of muscle, meaning that it could use fats and other molecules, including short-chain fatty acids, for metabolism. And the muscle would also be able to take up higher amounts of glucose, so it would be more insulin sensitive. And it would affect also the endurance of muscle. So these are probably traits that are also very interesting for agri-production and animal health. This is an overview of work package 5. So initially we had a high fiber diet. So Francesco's in the seminar as a nutritionist, and he helped design these diets based on what was known about short-chain fatty acids in the feces of pigs, fed different diets. We also had a high fiber diet, but included some sodium acetate in the diet. This would, short-chain fatty acids have been included in the diets. Acetate has also been included in human studies on insulin sensitivity in human subjects as a drink, and in mouse studies included in the water. And then we had group 3, which is, it says no fiber, but it's very low non-digestible fiber. So it would not be feeding those microbes in the colon that produce most of the short-chain fatty acids. And 2 months post-weaning, then in pregnancy 2 months post-weaning, we had 2 males, 2 females from each of the litters for making the biorepository. And in a second round or second parity of these cells, which remained on those diets, we had fetuses, 2 males, 2 females from day post-fertilization 70, also for the biorepository. The biorepository is a very interesting collection of tissues that we could look at for other effects of these short-chain fatty acids, but we focused on liver and skeletal muscle. Muscle because of the reasons I said, but liver also because it is an organ that metabolizes the short-chain fatty acids when they go from the portal vein, from the gut, and really reach high concentrations in the portal vein. They're metabolized by the liver. And it will be interesting to see what effects these have on liver metabolism and liver function on some of these other pathways, like lipogenesis, for example. So we have generated from liver and skeletal muscle, both from the piglets and from the fetuses, RNA-seq and ATAC-seq, and are now looking at the data analysis and integrated analysis. So what I can tell you about the diets is that we've had difficulties with technically measuring short-chain fatty acids in serum. They are not insurmountable, but technically that's taking us a lot longer to do. But we have measured short-chain fatty acids in feces recently, and we saw that these diets generally have the expected effects. So we will get data also on individual cells, and also from the piglets, which can be used for the data analysis and integration of that data. So we can stratify those, the piglets somewhat. And the high fiber plus acetate generated the most acetate in the feces, the high fiber second, and the no fiber a lowest amount of acetate. These differences are about 2 to 3-fold, and yeah, they are at levels which in humans and in mouse studies have had physiological effects. So we hope, yeah, in the future to see more from this. So the take-home messages are that yeah, short-chain fatty acids are major players in the interplay between diet, microbiota, and health. From a nutritional point of view, there may be apart from bulking, there is not so much attention, I think, given to non-digestible fiber, but it does produce short-chain fatty acids that provide energy for the enterocytes and important functions in the gut, but then they go beyond the gut and impact on other organs, particularly acetate, which is quite high levels in the serum. From some NMR analysis, we see that, so NMR is a very laborious technique. It's too laborious to use for all the samples we have from serum. But we see that the acetate reaches concentrations over 200 micromole per mil of serum, and propionate in the order of 20 micromolar, and butyrate at the very low micromolar, which is actually quite similar to what's been seen in humans who are on high fiber, let's say healthy diets, eating lots of vegetables and other sources of fiber. So we've seen that the short-chain fatty acid concentrations in the colon can be modulated by inclusion of non-digestible fibers that get fermented. And we hope to have some more news about the outcome of the data analysis in the remaining part of the project. And don't forget to feed your own intestinal symbionts too, because it's undoubtedly important for human health. Lastly, I like to acknowledge all the people involved in this study. So Elisabetta is the coordinator of GeneSwitch, along with Hervé as a deputy coordinator, but Elisabetta is also helping me very much with this cooperation in Work Package 5 and the running of Work Package 5, along with Hervé. And the team at INRAE, Sarah, Andrea, Shmana, and at Wageningen, it's Linda Lonen, Jos Buchholz, our bioinformatician, who's working with the bioinformaticians at INRAE. And also I'd like to thank Francesc Molist and his colleagues at Scott Horse Food Research, who were involved in running the animal study for us, and the collection of tissues along with Linda from our group. So thanks very much for listening.
Thank you, Jerry, for the interesting presentation. We have already 2 questions in the chat box. Okay. So the short chain fatty acids are microbial macronutrients, says Yap. Can microbial micronutrients be as important or even more important?
Yeah, I mean the vitamins, of course, that are produced by the microbiota that are important for us. Vitamin K, for example. Yes. So that's the example I know of for micronutrients.
Mm-hmm. And Reza asks, what is your opinion about adding short chain fatty acids on diet and then following its effects?
ask me what the opinion is.
Yeah.
I think it's a very interesting idea to also include them in the diet. The advantage of including them in the diet is this, that in order for short chain fatty acids to get transported, they have to be transported across the epithelium. Passive immunization is very slow in the colon. So we've tested this, in organoids, and they use transporters. But butyrate mostly gets metabolized. Propionate is transported, and acetate is the most highly transported short chain fatty acid. If you give it in the diet, in the stomach, they will be proteinated, and I think the transport will be higher. So I think drinking water or in the diet as a sodium salt will result in higher uptake of short chain fatty acids into the circulation. I mean, in the lamina propria, it happens and they go into the circulation because also the blood vessels there have transporters for this, but they have been little studied. But the transporters in the epithelium itself in the gut have been fairly well studied, but not in pigs yet. So I think it's a very interesting idea, and it may actually be an alternative to giving non-digestible fiber, but I think you might not get the same health effects in the gut, which are also very important for the overall animal health. And using fiber probably supports a healthy microbiome and other metabolites as well that are beneficial for health, like the aryl hydrocarbon receptor agonists. But maybe Francesc, if he's in the audience, could say what he feels about the use of fiber in diets and the circular economy and so on.
Thanks, Jerry. But no, I agree with you. In this case, we were using the sow. That is an animal that usually we can incorporate already quite a bit of fiber in these diets, and the sow will benefit from the fermentation. So that's why, yeah, this trial was interested also to use the sow as a model to see, okay, when we add, in this case was sugar beet pulp and pea fiber, and also we add some acetate directly to the diet to see what levels we can reach when we go to really high levels, because fiber will be a more common feedstuff in swine diet, as Jerry mentioned, because we need to use more byproducts from the food industry. So in this regard, it's also interesting to see when you go to these high levels of fiber, what you get back at blood level, and how the sow can use this fiber. So totally agree with Jerry that fiber will become more and more important.
Yeah, and certainly in the human area, I mean, there are huge meta-studies showing a correlation between the intake of fiber, the fiber that reaches the colon and is fermented, and all kinds of diseases, including cancer and so on. So there's actually very good metadata that healthy diets containing lots of fiber have many health benefits for humans. And although the pigs are not so long living, I think some of those health benefits, well, in the agri-production industry, I mean, some of those benefits will be short-term as well, on gut barrier, gut health, resistance to infection, immunity as well, a development of immunity in young animals.
Mm-hmm.